The Department of Veterinary Parasitology, College of Veterinary Science, Guru Angad Dev Veterinary and Animal Sciences, University Ludhiana, came into existence in the year 1975. The department imparts teaching, research and extension activities in the disciplines of helminthology, protozoology and veterinary entomology. The department also offers diagnostic services to the livestock owners, farmers and veterinarians related to the parasitic diseases through examination of the samples collected from the sick animals. Parasites are major constraint for sustainable and profitable animal production system. They cause huge economic losses, particularly in the tropical and subtropical regions. Broadly, these parasites can be categorized into three major categories: the helminths, arthropods and the protozoa. The helminths are further classified into three classes: the trematodes, cystodes and the nematodes. The trematodes are dorsoventrally flat, unsegmented helminths, commonly known as flukes. The cystodes, they are dorsoventrally flat, segmented, tape-like worms, commonly called as tapeworms, whereas the nematodes are cylindrical with tapering ends, commonly called as the roundworms. In arthropods, among the five major classes, the two classes Insecta and Arachnida consist of most of the arthropods affecting livestock health. Insecta includes insects like flies, fleas, bugs, lice, etc. Whereas in class Arachnida, ticks and mites are the major members affecting the animal health. Among protozoans, three broad categories are there: enteric protozoa, tissue-dwelling protozoa and blood protozoa. Enteric protozoa are the ones which are present in the gastrointestinal tract, like the coccidia, giardia and entamoeba, whereas tissue-dwelling protozoans like sarcosis, neospora, toxoplasma, and the blood protozoans include Babesia, Theileria and Trypanosome. The aim of the current video is to present an insight on the diagnosis of parasitic diseases. Clinical diagnosis in majority of the parasitic diseases is very difficult because of lack of pathognomic or disease-specific symptoms. Most of the symptoms exhibited by the animals suffering from parasitic diseases are very generalized, non-specific. Hence, laboratory diagnosis becomes very important for the confirmation of suspected cases of parasitic diseases. The laboratory diagnosis includes examination of various excretions and secretions like blood, feces, urine, lymph node aspirate, skin scraping, etc. collected from the suspected animal. So firstly, we start with the fecal sample examination. Diagnosis of gastrointestinal parasitic infection is done by the fecal examination. Most of the helminths and the coccidian parasites, they are present in the gastrointestinal tract of the host, and the laboratory diagnosis of these gut-dwelling parasitic infections is generally done by the demonstration of the developmental stages of these parasites in the fecal sample. Stages like ova, oocyst, cysts or even the trophozoites, which are generally present in the fecal sample. Collection of fecal sample constitutes a very important step which has a great impact on the result of the test. Hence, it needs to be carried out very carefully. Always, fresh fecal samples should be collected for fecal examination. And the best way to collect a fecal sample is to directly collect it from the rectum manually. And it is done by the use of lubricated disposable hand gloves for small and pet animals, whereas disposable sleeves for large animals are used. Vaseline or paraffin oil is generally used as a lubricant for the collection of fecal sample. Sometimes fecal samples cannot be collected directly from the animal and has to be picked from the ground, preferably pakka floor. In that case also, it should be always kept in mind that the sample should be fresh and it should be free from contaminants like urine, soil and dirt. Also, during collection of fecal sample, the portion of the fecal sample with mucus alone or with mucus and blood should always be preferred for collection, because this part of the sample more often inhabit the young or developing stages of the parasite in huge number. If the sample collected needs to be evaluated for the detection or presence of protozoan trophozoites, like in case of Giardia or trichomonad infection, it should always be examined immediately within 30 minutes of collection. However, in some conditions where storage of fecal sample is required, as they cannot be examined immediately within a couple of hours of collection, the sample should be refrigerated for later examination. However, the fecal sample should never be frozen, because freezing mostly distorts the shape of the parasitic eggs. The preservation of fecal sample is done by adding equal volumes of 10% formalin to the fecal sample. Fecal examination. Fecal examination is carried out in two broad categories: macroscopic or gross examination and the microscopic examination. The macroscopic examination or the gross examination of the fecal sample, it is usually done before the microscopic examination to look for any change in the color, consistency, smell, or simultaneously to look for the presence of parasites or stages of parasites in the sample. The fecal sample should be examined grossly for the detection of live or dead worms or segments of tapeworms. In ruminants infected with Bonnesia, typically cooked rice grain-like gravid segments are passed in the feces of infected animal. Similarly, dogs infected with Dipallidium caninum pass out typical cucumber seed-shaped gravid segments of Dipallidium caninum. Whereas diarrheic feces of ruminants infected with amphistomes and suffering from immature paramphistomosis exhibit profuse diarrhea, and the diarrheic feces reveal pink-colored immature amphistomes resembling pomegranate seeds. Similarly, the adult ascarid worms, yellow ascarid worms, sometimes are also passed out in the feces of the infected animals. Also, the information like the quantity, consistency, color, smell, presence of blood and mucus in the feces should also be recorded during the gross examination. Hookworm infection in dogs result in tarry, dark-colored feces because of the hemorrhage being caused in the anterior part of the intestine. Whereas whipworm infections result in diarrhea with excess mucus and frank blood coming out in the feces because of the presence in the posterior part of the intestine. Histomonas miliagridis infection in poultry causes sulfur yellow-colored droppings. And in Toxocara vitulorum infection of calves, mud-colored, foul-smelling diarrhea is seen with steatorrhea. Microscopic examination, and for the microscopic examination, the materials which is required consist of the containers for the sample collection, pestle and mortar, flotation solutions generally consisting of saturated salt solution, Seether's sugar solution, normal saline solutions, microscopic glass slides and cover slips, strainers, other glasswares, and most importantly a compound microscope. The microscopic examination is further classified into two broad categories, a qualitative and quantitative examination. First we'll talk about the qualitative examination. Qualitative examination is conducted to determine the presence or absence of the parasitic stages in the fecal sample, and it is generally done by two methods. One is direct smear method and other is the concentration method. We'll talk about the direct smear method. In this, generally what we do is a small quantity of feces is placed on a clean microscopic glass slide and it is mixed with a drop of saline or water. This slide is then tilted slightly in order to push the large particles or the debris on one side, and then a cover slip is placed on the suspension. The slide is examined directly under the low magnification of the microscope for the detection of the parasite stages. This is a very suitable and rapid technique for detection of helminthic stages in the fecal sample, but it has an inherited disadvantage of low sensitivity. That means it fails to detect low-grade infections, and this method is not very reliable, and that's why at least 3 slides should be examined before reaching a diagnosis. In concentration technique, these methods can detect even light infections and also save time by concentrating the eggs in a small volume. The concentration technique exploits the physical characteristics like the specific gravity of the egg for the concentration in the feces. It includes like simple sedimentation method. The principle of simple sedimentation method is like the specific gravity of water is 1, but that of the eggs of helminth parasites is always more than 1. So, eggs being heavier than water sink to the bottom and gets concentrated. This method is very good and useful for detection of heavier eggs like the trematode and some cystode eggs. The protocol for simple sedimentation method: approximately 1 to 2 gram of fresh fecal sample is placed in a mortar and little quantity of water is added before triturating it with the vessel to make a suspension, and if required more water can be added to make a good suspension. The suspension is then strained through a sieve and the mixture is transferred to a beaker. This mixture is placed in the beaker for at least 10 to 20 minutes to allow the heavy eggs to settle down. Then the supernatant is discarded, transferring a drop of this sediment to a glass slide and placing a cover slip over it. And the sediment is examined under the low magnification of a microscope . Simple flotation method, also known as the Willis technique. The principle for this is like the flotation solution used in the simple flotation method has a specific gravity much higher than that of the helminth eggs. Hence, it allows the eggs to float and get concentrated. The specific gravity of water is 1, and this specific gravity of water can be increased by adding impurities to this water in form of salt or sugar. Generally, saturated salt solution or sugar solution is used for simple flotation technique. This method is very useful to detect comparatively lighter helminthic eggs present in the feces, which consist majority of the nematode eggs, the strong eye eggs, and also some cystode eggs. Approximately 1 to 2 gram of feces is placed in a mortar and a small quantity of saturated salt solution is added to it. With the help of the pestle, it is mixed and made a suspension. If required, more quantity of saturated salt solution could be added to reach the required volume. Then this suspension is strained through a sieve and the mixture is transferred to a clean, dry vial and it is filled until a convex meniscus is formed. A clean cover slip is then put over the top of the vial so that it is in contact with the liquid and it is kept undisturbed for at least 10 to 15 minutes. Then this cover slip is gently picked up and placed on a glass slide and examined under low magnification of the microscope. Among the important parasitic eggs, the trematode eggs or the fluke eggs include amphistomes, dicrocilium, fasciola, cystosome, and all these eggs, they are typically operculated heavy eggs, generally encountered in sedimentation technique. Tapeworm eggs include egg packets of Dipallidium caninum, Tinea eggs, Monesia benedeni and Monesia expensa eggs, and also Hymenolapis species eggs. The nematode eggs generally encountered are the thick-walled Ascaris sum, Toxocara species, Par Ascaris, Ascaridia gelli, and strong eye eggs, which are thin-walled, whereas strongyloides eggs, which have a well-developed larva when laid inside, just like the lungworm or the Dictyocolous eggs, and Spirocerca lupi also. Trichuris and Capillaria eggs, they have bipolar caps present. The important parasitic protozoans inhabiting the gastrointestinal tract includes the Giardia trophozoid, Balantidium coli trophozoid, Balantidium coli cysts, Coccidian oocysts, unsporulated as well as sporulated can be seen in form of Isosporan species and Eimerian species. The second aspect of the microscopic examination is the quantitative examination of the fecal sample. This is done to determine the magnitude or the severity of the infection, and it is measured in terms of eggs per gram, often referred to as EPG in case of helminthic infections, or oocyst per grams or OPG in case of coccidian infection in animals. For the conduction of this quantitative examination, McMaster egg counting technique is most commonly used. The McMaster chamber has two compartments. Each has a grid which is etched on the upper surface. This chamber is filled with fecal suspensions and the debris in the suspension is allowed to settle down and the egg floats to the surface where they can be easily seen and counted for the determination of the egg per gram. For this, we take 3 grams of fecal sample and add saturated salt solution to make the final volume to 45 ml. It is thoroughly mixed and then this suspension is strained through a sieve. The chamber of the McMaster slide is then filled with this mixture using a pipette or a dropper or a syringe, and the entire chamber should be filled, not just the area under the grid. If you see large air bubbles in the chamber, it should be removed and refilled. Then this slide is allowed to sit for at least 5 minutes before examination so that the eggs can float in this period. Then the slide is focused on the low magnification and the number of eggs only within the ruled areas are counted. If one compartment is counted, the total number of eggs in one compartment is multiplied by 100 to determine the eggs per gram. Whereas if both the compartments are counted, the average of the eggs of both the compartments is then used and multiplied by 100 to get the egg per gram. Blood sample examination. Commonly encountered hemoparasitic infections in domestic livestock and pet animals are theileriosis, babesiosis, anaplasmosis, ehrlichiosis, and hepatozoonosis. The clinical signs in majority of these hemoparasitic infections, such as the varying degree of fever and anemia, are very common. However, the predominant clinical signs like hemoglobinuria in babesiosis, lymphadenopathy in theileriosis, and jaundice in anaplasmosis are also observed. In case of trypanosomosis, the signs are highly variable in different species of animal, like corneal opacity and edema of larynx is seen in dogs, whereas circling movement and edema of dependent parts along with cachexia is seen in other livestock species. Therefore, the confirmatory diagnosis through demonstration of the parasite in the blood samples by microscopy is an essential part to provide a specific treatment to the affected animals. The hemoprotozoas may be either intercellular, like Trypanosoma evensi, Trypanosoma theileri, or intracellular, like Babesia, Theileria, Ehrlichia, and Hepatozoon. The materials required for microscopic examination of blood samples include blood collection vials, disposable syringes, needles, microscopic glass slides and cover slips, methanol, stains like Giemsa, Leishman, Wright, phosphate buffer solution, other glasswares and plasticwares, and a microscope. Collection of blood. Blood is collected aseptically in clean vials containing anticoagulant, which is generally EDTA at the rate of 2 mg per ml, from various sites in different animals, such as the jugular vein in large animals, the cephalic or the syphanous vein in dogs and cat, wing vein in poultry, tail in rat, orbital plexuses in rabbits, ear vein in cat, rabbit, monkey, and anterior vena cava in pigs. For detection of Trypanosoma evensi, cerebrospinal fluid or the aqueous humor of the eye in case of dogs can also be used. Further, for the detection of schizont stage of Theileria annulata, that is the cock's blue body, aspirates from precrural or prefemoral lymph nodes can also be collected and examined after staining. Collection of lymph node aspirate. Restrain the animal properly before collecting the lymph node aspirate. Clean the site of lymph node aspirate with a swab of alcohol or spirit, and sometimes we even need to shave the area if required. Inject a small quantity of normal saline solution into the lymph gland with the help of a sterilized hypodermic needle. Draw the plunger of the syringe back to withdraw the lymph juice and transfer the aspirate on a clean microscopic glass slide and draw a smear. Blood smears it is essential that the slides to be used in making blood smears should be always unscratched and clean. The first type of blood smear is wet blood film. Place a drop of blood in the center of a clean glass slide. Place a cover slip over it and examine it under low power of the microscope. This is a valuable technique for detection of movements of microfilaria and Trypanosoma evensi without staining and can be quickly done. Thin blood smears. A small drop of blood is placed at one end of glass slide. A spreader is then placed on the slide at an angle of about 30 degrees and it's drawn back to make contact with blood. The blood is allowed to run across the spreader and then the spreader is pulled rapidly and smoothly in forward direction. A thin and even blood smear is generally V-shaped with feather edges. Quickly dry the smear by waving in air or keeping at slide warming table and label it. Thick blood smear. Place a medium-sized drop of blood on the center of the glass slide. Spread the blood with a toothpick or the corner of another slide. Allow the smear to dry in the air. Thick smear should be laked, that is, you need to dehemoglobinize it before staining. And this can be done by placing the air-dried slides in distilled water until the color disappears. Among the various stains used, Romanowski stains are most commonly used for staining of the blood smears and include Leishman, Wright and Giemsi stains for staining the blood smears. However, Giemsi stain is generally preferred for staining of hemoparasites. The principle of Romanowski stains: these are neutral stains that contain methylene blue and eosin. The methylene blue is a basic dye that binds to the acidic nuclei of the cell and imparts a purple blue color, whereas the eosin is an acidic dye which binds with the alkaline cytoplasm and gives it a red color. Giemsi staining protocol. First step in Giemsi staining is fixation of the smear. Fix the air-dried blood smear by dipping the smear in methanol or by putting methanol over the smear on the staining rack for 30 seconds to 1 minute. Air dry the smear. Prepare working solutions of the Giemsi stain from the stock solution by mixing 1 ml of the Giemsi stock solution with 9 ml of phosphate buffer solution with a pH of 6.8 to 7.2. Put this freshly prepared working Giemsi solution over the fixed smear and allow it to act for 40 minutes. Wash the stained smear under the tap water, air dry it and examine under the oil immersion lens of the microscope. Leishman staining, the protocol: flood the air-dried blood smear with Leishman stock solution for 1 minute. Add double the quantity of distilled water or buffer on the slide. Mix it well by a pipette. Allow this diluted stain to act for 15 to 20 minutes. Wash off the stain under tap water. Allow the stained slide to dry and examine it under an oil immersion lens of the microscope. Right staining. Add a few drops of right stain stock solution on the air-dried smear for a minute. Add equal drops of buffer on the glass slide. Mix it gently without touching the smear with the help of a pipette. Allow the diluted stain to act for around 2 to 4 minutes. Wash the metallic film with buffer or tap water and dry in air. Examine under oil immersion lens of the microscope. The microscopic examination of these stained blood smears. The stained slides are carefully examined under the microscope for intracellular parasites inhibiting the red blood cells like Filaria annulata, B.V.Sc A.H bigemina and Anaplasma marginale. Generally, the feather edge of the smear is preferred where the erythrocytes are segregated. Whereas for parasites like Trypanosoma evensi, Ehrlichia canis and Hepatozoon canis, the monolayer of the stained smear needs to be examined. Buffy coat examination. In case of chronic infections with low parasitemia, particularly in trypanosomosis and canine ehrlichiosis, smears prepared from the Buffy coat is considered ideal. Protocol. Blood collected in an anticoagulant is taken in a microhematocrit tube. One end is sealed. Carefully place the capillary tube in the centrifuge machine with the sealed end in the center. Centrifuge it at around 4000 rpm for 5 minutes. The centrifuged blood has the following 3 layers. Uppermost yellow layer, the plasma. Middle white layer, the white blood cells. And the bottom red layer of the red blood cells. Break the capillary tube at the junction of middle white layer of the white blood cells and the bottom red layer of the red blood cells. Transfer the Buffy coat contents on a microscopic glass slide. Draw a thin smear. Fix it with methanol. Quickly air dry it and stain with freshly diluted 1:10 dilution of Giemsa stain solution for 40 to 45 minutes. Wash it and air dry it. Examine it under an oil immersion lens of the microscope. The examination of skin scraping for diagnosis of parasitic mites. Parasitic dermatitis or mange is a condition caused by the infestation of mange mites, including Sarcoptes, Sauroptes, Demodex, etc. The condition of mange is generally characterized by severe irritation, itching, rubbing of the infested parts of the body against hard object. The lesions include alopecia or hair loss, dry, rough and wrinkled skin, accompanied by the presence of scab and foul smell in some cases. Some mites burrow into the host epidermis like Sarcoptes, while others spend their entire life cycle on the skin surface like Sauroptes. Most mites are microscopic, therefore skin scrapings are taken from the suspected animals suffering from mange or parasitic dermatitis for the confirmatory diagnosis. Collection of sample. For collection of skin scraping, the animal is restrained properly. Thereafter, the hairs in and around the affected area of the skin showing lesions of mange are clipped. Firstly, clean the scalpel blade by wiping it with tissue paper and dip its blade in mineral oil, and then scrap the skin several times in the same direction. The scraping should be preferably taken from the periphery of the lesions with a scalpel, as the mites are more likely to be present on the periphery as compared to the center of the lesions. For collecting samples suspected for demodicosis, the affected area is squeezed between the fingers, and the material oozing out should also be collected along with the scraping. And then transfer the scraping from the scalpel blade into a vial containing 10% potassium hydroxide or sodium hydroxide. For detection of ear mites in dogs, cats and rabbits, the animal should be gently restrained. Then a cotton swab is introduced into the external auditory canal and gently rotated. This swab is then placed on a piece of black paper and examined by a hand magnifier lens for detection of mites. An electrically illuminated otoscope cone may be introduced directly into the ear canal for detection of the ear mites, and in such cases the microscopic examination is unnecessary. Materials required for skin scraping examination: a hot plate for warming the scraping, 10% KOH solution used for digestion of the scraping, petri dishes, microscopic glass slides, cover slips, a centrifuge machine and a microscope is required. Examination of the sample. The collected skin scraping may be examined directly under the stereoscopic microscope, but generally the material collected is too dense for direct examination, and hence it should be brought just to boiling in 10% KOH or NaOH solution, or should be kept in this solution overnight at least to break it up. Once digested, the following methods are used for examination of the digested sample. Direct smear method. Transfer a few drops of the digested material on the glass slide, cover it with a cover slip, and examine under the low power of the microscope. Sedimentation method. Transfer the treated material in a centrifuge tube and centrifuge till the sediment settles at the bottom. Gently discard the supernatant and make a smear from the sediment and cover it with a cover slip. Examine it under a low power of the microscope. The important main mites generally encountered during the skin scraping examinations are Demodex canis. The mite lives in the hair follicles and sebaceous glands of various mammals, causing demodecting or follicular mange. Sarcoptes scabii, the mite which causes scabies in man and sarcoptic mange in animals. Sauroptic species, which are surface dwelling non-burrowing mites,